Now at lecture number 9 on patterns of inheritance, also known as Mendelian genetics. Here's basically what we're going to cover in this lecture. We're going to talk about different ideals of what inheritance means, and just briefly we're going to cover that in a few slides. Then we're going to go over Father Gregor Mendel. He is the guy around which this entire lecture is based, and he did most of his studies of genetics and inheritance using pea plants. We also are going to talk about alleles and what alleles truly are. Hopefully you've read the chapter by now and you have some inkling of what alleles are, but we're going to define them for you in a few slides. We're going to talk about the difference between genotypes and phenotypes, and we're also going to then conduct some hybrid crosses. We're going to take plants that have one kind of trait and we're going to cross them with plants of another kind of trait and see what the outcomes are. Those come in two varieties, at least that we're going to do for this class. You are responsible for knowing how to do monohybrid crosses and also dihybrid crosses. When we get to it, you'll know exactly what monohybrid and dihybrid means. Then we're going to, after we are kind of experts in how to do crosses, we're then going to learn a little bit about how genes interact with each other and how they might influence the outcome of what you might actually see in offspring. As far as traits go. And then we're going to also talk about environmental factors and how they might change the outcome of traits. So let's go ahead and start with some observations. This is a pretty typical observation. You know it to be true. Anytime you look at children, it's probably pretty easy to pick out who their parents are just by looking at similar traits. We have right here in this example, three ladies, right? We have the grandmother, the mother, and the daughter, and it's pretty darn easy to see that they are actually related. Same thing here. This, by the way, for those that don't know, that's the actress Sissy Spacek and her two daughters and her husband. Their daughters really are, it's really easy to see that they possess traits that are both from their mother and their father, and I'm guessing that's probably true of you too. Some of you have children, and I'll bet people tell you all the time, oh, your child looks so much like your spouse, but they have your eyes or your hands or whatever it is. There is something about the traits that we possess that is heritable, that we can actually give off to our offspring. The question is, if traits are getting passed on, how do these things actually get passed? What is the mechanism by which things that are outward appearance, your hair color, your height, your predisposition to certain genetic conditions or diseases, how is this inherited? Well, we had some pretty early ideas about inheritance. And what I mean by early ideas, I mean before about 1860. Before that time, people really thought it had to do with the blending of characteristics of you and your spouse, for example. Now, we weren't totally naive back then. We knew it had something to do with sperm and eggs and sex, but by the very act of inseminating or passing one seed, you ended up with a baby, and that baby ended up taking on characteristics of the two parents. The whole purpose behind this blending inheritance idea was that you take some essence of the mother and some essence of the father. You put them together, and the baby that you get was some intermediate between the mother and the father. These flowers here are a really good example of that, where you have a red flower and a white flower, and you mate those two flowers together and you get a pink flower. Well, that's what happens in nature. Well, that's an example of blending inheritance, right? White and red make pink. However, there are problems with that. You see, when you then turn around and take a pink flower and you mate it with another pink flower, you would expect by this blending inheritance idea that your offspring would all be pink. And yet that's not what happens. What you end up getting out of that when you cross a pink and a pink flower, red, white, and pink flowers. Well, that doesn't seem to be blending inheritance, so something's going on there, right? You would expect that if blending inheritance was the way things worked, that all variation in nature would eventually disappear, that you would always just get to the intermediate appearance of things. And yet, variation persists. And this blending inheritance really stumped Darwin. He had no idea what was going on. He kept saying blending inheritance, and in fact he even wrote that into a lot of the works that he had done, including some of his treatises on human evolution. And yet, it didn't seem to work for him. He kept thinking it had something to do with blood and mixing blood from one to another, and you get this intermediate case. Well, it totally stumped him. And that was the one thing, of all of Darwin's contributions, that was the one thing that just threw him for a loop. He couldn't figure it out. The person who actually came up with the solution for how this worked was Father Gregor Mendel. He was a Czechoslovakian mathematician who also happened to love gardening. Now we ascribe his skill set to gardening. However, what he was formally trained as was a mathematician. Now the fact that he was a mathematician who loved to garden turns out was a really good thing for us. Because he's trained as a mathematician, he understands a scientific approach, and he understands the whole concept of being meticulous and designing your experiments properly. He also was very methodical. He kept excellent records. That was the mathematician in him coming out. And he was also somewhat open-minded, which was also kind of a virtue here. So you get all of these traits together, along with the fact that he happens to be a pretty darn good gardener, and now you have the makings of a potentially great experimenter. Somebody to think about here also is, if they were ever to make a movie about Gregor Mendel, I would love for them to choose Kelsey Grammer. I mean, don't you think that Kelsey Grammer looks just like Gregor Mendel? All right, well, I'll call Hollywood and you call Hollywood, and let's let them know together. Now, he was using garden pea plants. The nice thing about garden pea plants is that you don't necessarily have to rely on other plants around you in order for fertilization to take place. You can actually take a pea plant and self-pollinate it. You can actually have the plant pollinate itself. You're just using a different flower on the plant. And the nice thing about these pea plants is that it is something that you can control for true breeding. True breeding means that if you take a trait, like for example, pink flowers, and you keep mating it with pink flowers over and over and over and over again, the offspring all seem to come out as pink flowers. You never get any other variation. Likewise, if you start out with white flowers, and you mate a white flower with another white flower, all of their offspring end up being white with no other variation to it. So those are true breeders. You always know what the genetic outcome is going to be. So this is really nice. You can then take what you know to be a true breeding pink flower and what you know to be a true breeding white flower, mate them, and then see what the outcomes can be. So this is actually very nice. You can actually control for experimental cross-pollination. The other thing that's really nice about these pea plants, they grow very, very quickly and very easily. It doesn't require a huge amount of maintenance. And, within a matter of weeks, you have results. Here is how Mendel kind of did this. If you want to mix flowers together, but you want to kind of control for it, what you can do is go into the flower and you can cut off the anthers. The anthers are actually the male part of the plant, or of the flower. So you cut off the anthers, and then you basically set it up so that this flower cannot accidentally inseminate itself, and instead you can take a paintbrush and take a little bit of pollen from some other flower that you specifically want and paint it onto the stamen of that flower so that you can directly control the reproduction. Another reason why Mendel chose to use pea plants in his experiment was that they possessed traits that were easy to identify, easy to recognize. For example, seed shape. Seeds were either smooth or they were wrinkled. Seed color. They were either yellow or they were green. The pod shape, so the ones that the seeds actually fit into, they were either smooth or they were corrugated like this. Pod color, either they were green or yellow. Flower color, white or pink. Flower position, are the flowers at the end of the stems or are they at the base of the stems? And then the stem length. Is this a really tall plant or is it a very short plant? So these 7 traits here were very easy to track, and Mendel really liked this. So what did he end up finding? Well, he began with true breeding plants. That is, he bred these things together and they always ended up producing the exact same offspring. So, for example, using smooth yellow seeds, or in this case I have wrinkled and yellow, but that's not what I'm showing you over here. Let's say you start out with wrinkled seeds, and every time you breed wrinkled seeds with other wrinkled seeds, that's what you get, always, 100% of the time, wrinkled seeds. Same thing with yellow, 100% of the time you get yellow every time you try and cross a yellow with a yellow. Well, then you know that those are true breeding. I've given you an example here of a smooth yellow pea plant, and every time you reproduce it, it always spits them out 100% of the time as being smooth and yellow. The trick is then to take these true breeding plants and crossbreed them with other true breeding plants and then examine the offspring and see what actually happens. So there's a couple of terms that we have to kind of define here. The first one here is the parent generation. What are we talking about when we refer to parent generation? We mean the first generation that when we're just now getting ready to cross. So you take one true breeder, you take another true breeder, and you cross them together. They are the parent generation. The parent generation then mates to produce the first generation offspring. The first generation offspring are also known as the F1 generation. That's F as in Frank. You can then take members of the F1 generation, mate them together, right, to end up with a second generation offspring. We call those the F2. Another way to look at this, by the way, visually, would be like this. We're going to take true breeding yellow seeds with true breeding green seeds, and we're going to cross them together. They become the parent generation. Okay? And don't worry about these terms over here. We're going to bring these up a little bit later. We start out with our true breeding plants here, and we're then going to mate them together, and lo and behold, when you mate a green and a yellow seed, 100% of the offspring in the F1 generation are yellow. However, when you take that yellow seed and you mate it with another member of the F1 generation, another yellow seed, you end up producing yellow and green seeds. Well, that's kind of weird. And not only do you produce yellow and green seeds, but you always seem to produce them in a ratio of 75 to 25, or 3 to 1. That's a 3 to 1 ratio. A quarter of the offspring tend to be green, and 3/4 of the offspring tend to be yellow. So this is his first observation here that totally stunned Mendel. When he did this parent to F1 to F2 breeding experiment, no blending inheritance actually occurred. You end up getting 2 colors back out in the F2 generation. Well, that's kind of weird. It means that the traits that are inherited by the offspring are some sort of separate units. He didn't quite know what that meant at the time. He just knew that they were kind of separate. That you take a yellow and a green, you mix them together, that is, you make them mate, and you end up with all yellows. But when you take the yellows from that F1 generation and make them mate together, the F2 generation always spits back out both colors that you started with, but not in an equal ratio. So they're either yellow or green, which means he got both colors back in the F2 generation. Here's another way to look at it. Just another way here is that you have your yellow and your green that you're mating. Again, 100% of your offspring become yellow, but that when you take some of these guys here in F1 and you mate them together, you end up with 25% of them being green again. Now, since it's 25% is green and 75% is yellow, Mendel decided to come up with a name for this. He said that yellow is dominant. He came up with this determination that yellow is dominant because in the F1 generation, all of them are yellow, and then in the F2, most of them are yellow. Okay, it makes sense. He called yellow the dominant one. Now, when we refer to dominance, for the rest of this lecture. The things that are dominant, we're going to give them capital letters. So in this case, it would be capital A. Capital A refers to dominance. Now, what about the other case here, that green? Remember, green is the one that disappears in F1 and then comes back in F2 at a smaller ratio. Well, we call that recessive. It's recessive because we don't see it in F1 and it reappears in F2. And we give the recessive trait a small letter A. And so this will help us distinguish between dominance and recessive. Yellow is dominant, and the green is recessive. And we give them the same letter. This is A and this is A, but this is just capital A, and this one over here is small a, implying that A simply refers to pea color, okay, pea plant color. So green is recessive. All right. Now, this is true actually in you and your offspring, and also from your parents. You have 2 sets of DNA. We've actually already brought this up before. You got 23 sets of DNA from your mom, 23 from your dad, and that's where you got your total of 46. And little Maggie here from The Simpsons acquired her DNA from Marge over here and from Homer. So that's kind of what's going on there. And this is where your genes are. This is where all of the DNA instructions on how to make you resides. Okay, now you know this to be true. I just wanted to remind you of this. Mendel did not know about this because the whole concept of DNA did not exist at that point. So here's Mendel's model. Here are the 4 basic concepts in Mendel's model. Number 1, there can be alternate versions to any trait or any gene. For example, in pea color, it can be either yellow or green. Nothing else, just yellow or green. The flowers come in alternate cases, either purple or pink, and white, either pink or white. So these are alternate versions. And number 2, you only get 2 alleles. That is, you can only get one from mom and one from dad. You never get 3, you never get 12, you only ever get 2. If 2 alleles are actually different in your DNA, then one of them plays a dominant role. That is, one of them gets expressed and the other does not. Now, the 4th rule here is this law of segregation, where 2 alleles end up getting separated in gamete formation into sperm and eggs. In other words, in your body, let's say that you have parents who have different colored eyes. Let's say your mom has brown eyes and your father has green eyes, like my parents actually. And so I carry both of those alleles. I carry the allele for green eyes and the allele for brown eyes. However, brown is dominant over green, so my eyes happen to be a shade of brown. Okay, but I still am a carrier for green, and so the sperm cells that are being formed in my body and the sperm and egg cells that are being formed in your body end up getting separated out, and so one of them can actually, or half of them can carry the gene for green eyes, while half of them can carry the gene for brown eyes. So that's why, even though I exhibit brown eyes, it's perfectly likely that my offspring could have green eyes if the person I mate with has green eyes in their genetic history. Okay, so that's what the law of segregation really refers to. And now that whole Homer and Marge Simpson idea is true with pea plants too. So now we're bringing it up to the present here. The same thing is true in pea plants. You can have green and yellow, and when you cross a green and yellow pea, the DNA that accounts for that yellow and the DNA that accounts for that green end up going into the offspring, even though the offspring can only express one of those traits. It becomes a carrier for the recessive trait. So here are some terms that you have to know. What about a gene? What is it that we're referring to? A gene is simply a unit of genetic information that gets passed on from the parent to the next generation, to its babies, its offspring. And individuals have pairs of genes. Remember, you get one from mom, one from dad, and they originate from the sperm and the egg that made you up. So the other thing is that traits tend to be either dominant or recessive. Again, you can think about it for hair color, for skin color, for height, for lots of other genetic traits that we're going to discuss a little bit later. Now, we now have to talk about this concept of alleles. I brought up two eye colors. I brought up brown and I brought up green. But actually there's more eye colors than that, aren't there? In fact, there's a whole bunch of shades of brown. There's hazel eyes, there's light brown, there's dark brown, there's blue eyes, there's gray eyes, there's green eyes, there's all different shades in between. And so even though I can only ever have two genes in my body to control for eye color, there are a whole bunch of other genes in the human population for other eye colors that I don't possess. These are all referred to as alleles. So alleles for eye color actually include dozens and dozens of different possibilities, of which I can only ever carry two of them. So an allele is a unique type of the exact same gene. It makes it really easy when there's only two types, right? For example, wrinkled or smooth. But it makes it a little bit more complicated, but just as likely, if I refer to green eyes, brown eyes, hazel eyes, blue eyes, and gray eyes. Those are all alleles as well for eye color. We're going to make it very simple here, though, in dealing with Mendel's P's, because in every one of these traits that we discussed, these 7 traits, they only come in 2 varieties, like, for example, yellow or green, or wrinkled or smooth. And since you have 2 copies of DNA, let's say this would be chromosome 1 from your mom, and this is chromosome 1 from your dad, in this same location here, you could have 2 different alleles that code for a slightly different thing. For example, let's say G refers to your height. Well, this is G height being short, and this is G height being tall. Okay? This might be hair color, and this one might be eye color, and this one might be something else. But you could possess more than one way to express that trait. And actually in each one of these examples, G, R, S, and T, they are different, right? There's capital and small. But it is entirely possible that you could possess, a capital T from your mom and a capital T from your dad, in which case there'd be no difference. That actually is the definition of a true breeder. Okay, then there's something called a gene locus. Locus is a scientific term meaning area or zone. So a gene locus is simply the area on the chromosome where a gene is located. Now, a couple of other terms to think about here. Each one of these chromosomes, remember, these are not different chromosomes. These are the same chromosome, chromosome 1, for example. But this is chromosome 1 from your mom, this is chromosome 1 from your dad, and so therefore we call these homologous chromosomes. Homologous does not mean identical, though, because remember, you could potentially have an allele over here that expresses green eyes, but over here, in the same eye location on this one, it's expressing brown eyes. Okay? Now, the dominant versus recessive thing, well, remember, when we're referring to dominance, we are simply saying that you may possess 2 alleles, but the one that is expressed is the dominant one. So I'm just going to make this up just to make this kind of funny. Let's say that G stands for grumpiness, where capital G would be grumpy and little g would be not grumpy. Okay, happy. There you go. That would be the happy gene over there. Big R means round, little r means square. Maybe big S means sweaty and little s means not sweaty. Okay, I'm totally making this up, guys. Okay, big T means tall and little t means the opposite of that, short. So let's say that this is our actual genetic combination. Who did we make? Well, I did my best to make a grumpy, round, sweaty, tall guy. Okay, I know that's kind of funny. I did the best I could. Alright, so what about allele combinations? Well, let's, keep this simple. We're referring to pea plants here, where we have either wrinkled or smooth, or yellow or green. And in this case, you have 3 different allele combinations that are possible. So I'm going to throw a couple other terms at you here that you do need to know. Homozygous. means the same as, and zygous refers to the genetic complement. And so homozygous, the condition of being homozygous, is where your alleles are identical. So you have two identical alleles at a locus. That is, your mom has brown eyes, your dad has brown eyes, so you have brown eyes, right? And in fact, if you look on the gene that codes for eye color from your mom and the gene that codes for eye color from your dad, they are the same. Okay, so you would be homozygous for that trait. However, I had mentioned before that brown eyes is dominant, right? So you would be homozygous dominant for brown eye color. However, let's say that you have a mother with blue eyes and a father with blue eyes. In that case, blue is recessive to brown, and so you would be homozygous recessive. In this case, little a, little a. Now, this can be for anything. We can refer to pea plants here as well. Now, when you have a big A and a little a, let's say you got brown eyes from your mother and green eyes from your father, or blue eyes from your father, well, they pass those on to you, and your condition is said then to be heterozygous. So this right here would be big A, little a. That's the condition of being heterozygous. So these are the three possible combinations that you could have for this one particular allele on your DNA. So you could either be big A, which is homozygous dominant, little a, which would be homozygous recessive, or big A, little a, which is heterozygous dominant. Why heterozygous dominant? Because big A is dominant over little a, and so big A is the thing that would be expressed in your outward appearance if you possessed this combination over here. Just to put into contrast the difference between homozygous and heterozygous. Hetero means different than, or opposites. So this is another good example of a typical gene in your body that is controlled by this same kind of mechanism. If you are big S for finger length, well then you are considered homozygous dominant. And what would end up coming out is you would have short fingers. Believe it or not, short fingers is dominant over long fingers. Okay, then you have big S, little s here. In this case, this would be heterozygous, right? If you are heterozygous, you're still going to exhibit short fingers because at least that dominant gene is present. Then you come down to here and you have little s, that is considered to be homozygous recessive, and a person with little s would end up with long fingers. Now, two other terms that I would like to bring up, and I'm going to mention them again a little bit later, is this genotype and phenotype. Genotype refers to this combination here of what you have in your DNA. Phenotype is the outward appearance of what happens when this is expressed. Again, I'm going to come back to this in a couple of slides. So, here's what we're talking about. If expressing recessive traits like little a, we automatically know what its genotype is. We know what its allele combination is. If you are a green pea, there's only one combination of alleles that you can have. You must be little a. You cannot be any other combination. But if both big A and big A, little a both express the dominant gene, if somebody is yellow, you aren't necessarily going to know what the genotype is. You just know that it has at least one big A. But this blank over here can be either a big A or a little a. There's no way for you to know that. Now, again, just to clarify what this means, genotype and phenotype, differentiating between these, a genotype refers to that allele combination, okay, which is shown right here, whereas a phenotype is that outward trait appearance that you see, yellow or green, wrinkled or smooth, etc., something like that. You cannot always know the genotype from the phenotype. It's not possible. Sometimes you can know, especially if it's a recessive trait. If you're exhibiting a recessive trait, then it means that your genotype is automatically known. And I want you to think about why that is, right? Because there's only ever going to be one combination of a genotype that would exhibit a recessive phenotype. So here's some good practice, okay? Let's go with these two seed colors. Big A is yellow. Little a is green. That is, big A, yellow, is dominant. Little a, green, is recessive. So, what is the phenotype? Let's think about this for a little while. The phenotype refers to green and yellow. What is the genotype? Well, in this case, you know what the genotype is, right? It has to be little a, little a. What's the genotype of this? Well, in this case, there are two possibilities, right? It can be big A, or big A, little a. You don't know. So you'd have to write both of them on the exam, unless I gave you a little bit more information. So now let's come back to this. How do you explain this, knowing what you know now about genotypes and phenotypes? Well, you've got your parent generation here, where you've got yellow and green. You're producing an F1 generation that is 100% yellow, and then in the F2 generation, the 3/4 of them are yellow, and 1/4 of them is green. So what's actually going on? In order to understand this, and why it is that these percentages here are always so precise, we need to go back to meiosis. That's right, everybody. Heredity has to do with meiosis, and once again, we come back to sex. That's just the way it is in biology. We talk about sex a lot. Okay, here we are with our parent population. When we're taking a parent generation, a yellow and a green pea plant that we're going to be mating together, what are we actually mating? Well, we're taking the equivalent of sperm and eggs. In this case, we're taking pollen and an ovary, right? But it's equivalent to like your sperm and eggs. And we are separating them into, we're taking their genetic complement from the adults, and we're separating them into the pollen and in the ovaries, like this. Well, we're going to take a wild type example first. So we're starting over here. You automatically know that that has to be little a, right? Because that's the only way to make green. And we know that this is big A because we've done a true breeding cross. We've mated this like 12 times in a row, and it always produces 100% yellow. So we're pretty sure that that's big A, and not heterozygous. Remember, you have 2 copies of DNA. This has 2 copies of DNA. And when you make sperm and eggs, or in this case pollen and ovaries, you are splitting those apart. Like that, and like that. Okay? So in this case, let's say this is you over here. Your big A and your big A end up going into the pollen, whereas this little a and little a end up going into the ovaries. So this is really important. This is Mendel's theory of segregation, and that is to say that an individual inherits 1 unit of information about a trait from each parent. So even though you possess 2 genes for something in your body, in the sperm or the eggs that you are making, only 1 set of those genes can go in. Again, I possess eye color for both brown and green, even though my eyes can only be one of those. When I make sperm cells, then both green-eyed genes and those brown-eyed genes get separated back out and stuck into those cells. So this is what happens. If you are a heterozygote case like big A, well then half of them are going to have the big A's and half of them are going to have the little a's, like this. Now, this is pretty interesting. It means that during gamete formation, alleles segregate from each other. And I want you to kind of refer back to meiosis to kind of remember how this happens. So let's take a look at the parent genotype. Can we predict the outcome of various crosses? Let's assume that you know the parent is big A. What can that parent contribute in its sperm or eggs? It can only ever contribute a big A, right? It can't contribute anything else. What about if the parent is little a? Again, for the same reason, in its sperm and eggs, it can only ever contribute a little a. However, if a parent is heterozygote and has a big A, then it will end up contributing either a big A or a little a to its next generation. So let's go ahead and actually conduct a couple of these crosses, and these are called monohybrid crosses. Okay, these are called Punnett squares, and yes, you are going to have to be able to do this on the next midterm, conduct a Punnett square. And Punnett squares are just these little squares right like this. And a mono-hybrid cross, mono means one, and hybrid meaning that we're only crossing for one trait here. So we're going to take two pea plants and cross them for this yellow-green thing here. And I want to use an obvious case here, something that's really obvious to start with. Let's say that we have a female gametes here that are both that are big A, and a male gamete that is big A here. So this would be the eggs over here, this would be the sperm over here, and we're going to cross them and look at all the potential outcomes. Remember, this male gamete, the adult, was big A, which means in its sperm, or in this case, yeah, in its sperm or in its pollen, it's only going to be big A's. Same thing with the female here. And when we cross them, this is what we get. In other words, 100% of the offspring are always going to be yellow. And this is, again, how we determine a true breeding species. If we made them over and over and over and over again, and 100% of the offspring are the same as the two parents, well, that's an example of a true breeder. But let's kind of shake things up here a little bit. What if we have this condition right here, where we have a female that is only producing big A's and a male that is only producing little a's? Well, what's going to happen? Let's go ahead and conduct those crosses. And now we have just made all of our offspring here, and they are all heterozygotes. Well, what is the phenotype? In this case, the phenotype is all yellow. Remember, yellow is dominant over green. So even though these are carriers for the green gene, they are only going to be yellow. That's the phenotype, yellow. How many yellows do you have? And how many greens do you have? Okay, I'm going to let you think about this. Well, in this case, 100% are yellow, 0% are green. But now let's take this. This is now the F1 generation. Let's take some F1 generations and mate them with each other, like this. So F2 cross, we're now taking big A, little a, we're crossing it with big A, little a here. These, again, are the potential genes that can exist in all of the eggs and all of the pollen of these plants. And if we cross them, look what we get. Interesting. This big A combines with this big A combines with this little a combines with this big A, and this little a combines with this little a. And we end up with this huge variety. What are the phenotypes? Well, take a look at this now. The phenotypes, even though you're mating things which phenotypically are yellow, only 3/4 of them here are going to be yellow. This one because it's homozygous dominant, but these two here are heterozygotes and they have big A's in them, so they're all going to be yellow as well. But look what you get. You end up with 25% of your offspring as being green. So how many are yellow? 1, 2, 3. 3/4 or 75%. How many are green? That was yellow, I'm sorry. 3/4 are yellow. How many are green? Well, in this case, it's 1/4 or 25%. So this is kind of another way to look at it. We're taking a true breeding plants here. In this case, white and pink flowers from these pea plants. And we're mating them. And we end up producing an F1 generation that is all the dominant type, okay, even though they're all heterozygotes now. And now we're going to take examples from this F1 generation and breed them together. And what we end up with is, so that's what we've done here, we end up with 3/4 of them coming out in F2 as pink, but 1/4 coming out as white. Now, you can do this, by the way, with a huge number of traits, and I'm going to show you a good example of this. It turns out that ear, whether you have a hanging ear or an attached ear, this is a really good example. Having a detached ear like this is actually dominant over having an attached ear. I happen to have a detached ear. So let's say that this is somebody that you know, and you're probably looking in the mirror right now to try and decide whether you have an attached or a detached ear. And let's go ahead and cross these people. Well, how many are from this cross are going to have hanging earlobes versus attached? Well, let's do the cross. Look at that. 100% are going to have hanging earlobes, even though the offspring are probably all going to be heterozygotes, just like this. But now, oh, and you take a poll in the class. How many of you have one type or another? I don't know. Maybe I'll put up a survey so that we can try and do this as a class. But now let's cross that F1 generation. Essentially what we've done is we've now taken the sperm and the eggs from the population, and we've stuck them into this Punnett square here. What are we going to get? Voila! We get 75% hanging earlobes, which includes this one homozygote dominant and then these two heterozygotes here. And we get 25% with attached. Isn't that amazing? So can you see answering a question like this on the exam? That would actually be quite nice. So guess what? This is what Mendel was observing the entire time that he's doing this experiment. He's doing all of these monohybrid crosses, crossing purple and white, crossing axial versus terminal flowers, yellow and green, all of these different things. And he performed these a huge number of times. You want to know how many times he crossed purple and white? Right there. He crossed them like 929 times. 929 crosses. And in 929 crosses, he got 705 that ended up coming out in the F2 generation as purple, and 224 coming out as white. And these are the ratios. Now, I keep saying like 3 to 1, right? 75%, 25%. Well, they weren't always exactly 3 to 1. Sometimes they were 3.15 to 1. Sometimes they were 2.96 to 1. Why are they not exactly 3 to 1, even though these are all pretty darn close to it? What it comes down to, why it's not exactly 3 to 1, is because every time you cross an organism, it's still all just a probability trial. When you only have 1 or 2 children, there's no rule that says it's not going to be the rarer case. But in order to get close to 3 to 1, you have to have lots and lots and lots and lots of babies. Think about flipping a coin. If you flipped a coin, okay, let's go ahead and picture that in your mind. In fact, go get a quarter and flip a coin. What are the chances it's going to land on heads or tails? It's 50%. There's a 50% chance it's going to land on one side or another. But does that mean that every time you flip it, it's always going to be one or the other? What if you flipped it 3 times in a row and you got 3 heads? Would you be surprised? Well, of course not. I mean, that's likely. You could possibly do that. How about 6 times? If you flipped it 6 times in a row, could you get all heads? Yes, probably. But the more you go, the less likely it's going to be. And if you did it 1,000 times, chances are about half are going to be heads and about half are going to be tails. Same thing here. The probability outcome should be somewhere around 3 to 1, but it may not necessarily be always 3 to 1. It comes down to large trials. The more trials you do, the larger the number of trials, the more accurate your number is going to be. So no matter how much this woman here is going to try and come out with some Punnett square, she's got, wow, 8 children here. She's probably going to have about 800 children in order for us to see that statistical difference. I don't recommend that. Okay, so natural systems are variable. Random chance affects gametes and offspring. It's always going to be random, even though the probability is going to predict that it should be 3 to 1. Now, when we think about this in terms of gametes, every gamete has an equal chance of receiving each allele. Remember, we start off with this in our DNA right now. Remember, you have 2 copies of your DNA. In each of these pollen grains over here, there should be a 50% chance that this big A or this little a is going to get inside of each one of these. Okay? And really what a Punnett square prediction is doing is it's saying we can know the genotype from looking at the phenotype only in certain instances, right? So in this case over here, when we have white flowers, little a, we know it has to be that way. For the pink flowers right over here, we don't exactly know what the genotype is going to be. We just know that it's one of these 2 as a possibility. Well, let's say you didn't know, and you were to take one thing that you did know and cross it with something that you didn't know. Could you predict the outcome based on this? The answer is probably. You see, here's what we're going to try now. Okay, you know what the genotype is of this. It has to be little a, or in this case, little p. We're just using a different letter here. This one can be either big P, or big P, little p. You don't know. So you're going to cross the white flowers with the purple flowers and see what you get. You see, if it was big P, then 100% of the offspring are going to end up being heterozygote, and they're all going to end up being this purple color. But if it was heterozygote, big P, then 50% of the offspring are going to be white, and 50% are going to be purple. So just by looking at the outcome of the F2 generation, or of the offspring generations, you can figure out if the results were this, then the parent up here must have been homozygous dominant. However, if it's 50-50, then this parent must have been heterozygote. So, using this concept, I decided to set out and do my own test cross. I mentioned that hanging ear thing. Okay, that's my ear. Yes, that's my actual ear. Notice that I have a hanging earlobe. That is my wife's ear. She was really thrilled that I took a picture of her ear for this slideshow. Notice that her ear is attached. I wanted to know if I was heterozygote or homozygote for a hanging earlobe. I knew that she must be little e, my wife. I didn't know if I was big E, or big E, little e. So I had this plan all along. I decided to have a child. Okay, I didn't actually have the child. My wife had the child, but, I kind of helped. And it turns out this is my daughter's earlobe. What do you think? Attached. Little Jade, my little daughter, has an attached earlobe, so what does that mean for me? It means that if I was homozygous dominant, my daughter would automatically have hanging earlobes. But she doesn't. She has attached earlobes. And the only way for my daughter to have attached earlobes is if I was heterozygote. So how about that? This is the only reason I had a child, because I always wanted to know about my earlobes. Actually, that's not true. Actually, the only reason I had a child is so that I had somebody to ride the roller coasters with me. Alright, what is the genotype of the unknown red parent in the offspring? You can kind of see how this would go if I was to provide this on an exam. You have the white one here, you automatically know it's little a. You don't know what this one is. You just know that it has at least a big A in there. You don't know if it's big A or big A little a. However, in showing you the offspring crosses here, the fact that you have 2 purple flowers and 2 white flowers means that there's only one choice. And what is that? That's right. It has to be heterozygote. It cannot be any other thing. That pink flower has to be big A, little a. Let's try another one. What about if you had big T, little t for tall versus short? What is the percentage of flowers that would be tall if you were to cross a big T, little t with a little t? You can pause it here and actually try this one now. And if you said 50%, then you got that one right. Okay, I'm going to actually break here, and we're going to then pick up the second half of this lecture in a second set. So I'll see you all in the next lecture.